So we'll move on to the next speaker, which is Alicia Paini. She's a member of the Cosmos Consortium as well, and it's going to be a slight shift. So we're going to talk about the other aspect that Elena alluded to in her introduction there, which is biokinetic. So Alicia is going to talk about in vitro to in vivo extrapolation, modeling to predict target organ concentrations. And I'll give you...
We have developed in order to perform in vitro to in vivo extrapolation to predict target organ concentration inside the human body. The outline of today's presentation will be in two parts. The first part is now. It will be this introduction, biokinetic models developed in Cosmos. After the coffee break, there will be a hands-on session where I will demonstrate how the workflows that were developed for this type of models could be applied in an automated way using the NIME platform. So I would like to start by giving a short introduction. So all of you know that we are daily exposed to chemicals. They can come from the environment, such as pollutants. They can be contaminants can enter our food chain. Even our food products contain sometimes natural chemicals which can affect our body. And also consumer products have a lot of chemicals which can be sometimes in high doses harmful to our body. Our intake can be with different routes, so inhalation, ingestion, or skin absorption. And once the chemical is entering, you have then the absorption throughout your body, distribution via the blood. Once it will reach the target organ, within the target organ, the organ can metabolize the chemical to metabolites in order to achieve chemicals which are more hydrophilic that can be excreted and eliminated, or they can actually become more harmful and make an effect. So the first part of absorption, distribution, and metabolism is so-called toxicokinetics, while when the chemical is interacting with the body, we have more so-called toxicodynamics. And this effect can lead to a cascade of events which can lead to your adverse outcome. So how can we apply safety assessment and in a risk assessment? So we have an in vivo, in the past we were using in vivo data from rodents, but now, especially for cosmetic ingredients, there is a European ban since 2013, so we are shifting more to in vitro data. So really the risk assessment has to come out from in vitro data and in silico computational tools. In order to especially fill gaps between in vitro and the in vivo situation, we can use computational models as well. So type of computational models that we can use are so-called physiologically based kinetic models, which represent the human and interconnected compartments. And we can say after an exposure to a chemical, how much of this chemical will reach the target organ, or what happens in vitro. So once you have your chemical within your medium, how much of this chemical is actually going to affect your cell? And this is so-called virtual cell-based assay. So all of these models are trying to reach the goal of the Cosmos to establish, let's say, prediction for long-term repeated toxicity to humans, and in order to predict target organ concentration. And of course, we're trying to develop these models in an automated way in order that the end user can use this in an easy way using the NIME software. Cosmos tools that we have developed were in several areas, so in absorption, in biokinetics, in chemical space analysis, and in structural alerts. For today, I will talk about the biokinetics. So as I just mentioned, PBK models and virtual cell-based models. We also have a human bioaccumulation model and multi-scale models. So physiologically based kinetic models are, as I just mentioned, models that are represented by differential mathematical equation. They're representing the human body as compartments. So for each compartment, you can simulate either an organ or the bloodstream. The chemical will enter, you will input, and it will, because of the differential equation, there will be the so-called simulation of the transformation of the chemical from a dose to a concentration inside the organ. And these equations are built with parameters, and these parameters are basically physiological, physiochemical, and biochemical parameters. And it's interesting because you can actually simulate time profile or dose concentration or concentration effect dose ranges in order to test the chemical and the effects of the chemical throughout the body. How did we build this model? So I think this is one of the interesting slides. So we use 6 steps. So theoretically, we followed the paper from Richens et al. From 2011. So you first define the conceptual model. So you have your chemical. You know what is the target organ. You structure, you design on a piece of paper your model. After you have your conceptual model, you can translate it into mathematical equation. So here we have an example of the absorption in time of the chemical. This is given by the uptake from the GI tract and the transport through the arterial blood to the venous blood, minus, of course, the amount that is actually metabolized. And all of these equations are defined by parameters, and these parameters can be either given by found in the literature, or you can look by measuring yourself in vitro, as well as looking at in silico prediction with QSARs. And these can be tissue volumes, blood flow rates, partition coefficient, uptake constants, and metabolic parameters. After you have all these set up, you have to solve. So you have to find a software which can solve for you the equation. Usually you can have Berkeley Madonna, MATLAB. For the Cosmos project, we try to use software which are open source, so freely available to download. So we use the R, and we use the Dissolve program. After everything, the script is written, and it can be used to simulate. You have to actually look at how it performs. So you actually need some, let's say, validation. In order to evaluate your model, you need in vivo data. Sometimes you can find them in the literature, sometimes you're unlucky, so you just try to estimate based on read across or looking at how other chemicals were performing. And the applicability of this type of models are, especially in the risk assessment, can be used to go from a high dose that is used in rodents testing to a low dose that is more, let's say, relevant for the human exposure, to go from a different route-to-route extrapolation, so to go from an absorption from the skin to an oral, from, let's say, oral, which is more common to the skin, and also to use from in vitro to in vivo extrapolation. It's used also to do acute and chronic exposure, and also from single to repeat exposure. So in the Cosmos project, we developed PBK models for all of them and inhalation, so for the 3 different types of intake routes, and for 10 different compounds that are listed in this slide. We are also trying to make a general model. It's ongoing this at the present moment, in order that actually if you have a chemical for which you don't know anything, you can just enter your parameters and you can simulate. The second type of biokinetic model developed was a virtual cell-based assay. And this model actually helps you to simulate the fate of the chemical within the laboratory well. So when you are in the laboratory, you have a 96 well plate, for instance, you see what actually is the fate of the chemical inside the small well. And also in order to simulate the concentration leading, the intracellular concentration leading to the cell perturbation or to the effect. The model consists of also differential equation. It's built up of 4 sub-models, so the fate model, the cell model, a cell growth model, and a toxic effect model. The fate model actually tells you what happens to the chemical when in the medium. It can either bind to the plastic, to the lipids, to the protein. It can evaporate to the headspace. And so a small amount will go, small amount will go actually to affect your cell. And this fate model is actually built up on different chemical properties, such as air constant, log P, air and water degradation, molar volume, and atomic diffusion. All of these in the model are based on QSAR predictions. The cell model actually tells you how much is uptake of the chemical within the cell, the diffusion in the cell between the 3 different compartments, so the water, the protein, and the lipids, and it's built up of partition coefficients and mass fraction of these compartments. Then we have a nice feature, which is the cell growth, which if you have a cell line in your in vitro testing which proliferates, you can also activate this part of the model, which tells you how much of cell cycle of the cell growth it can happen during your in vitro analysis. So it takes into account information on the cell cycle dynamics. And finally, we have the toxic effects, which actually give us the readout from the measurement of your in vitro testing, based on the so-called no effect concentration and killing rate in order to be able to perform so-called simulation for the final adverse outcome or effect that we can measure in vitro. Also here, the model equations are run in MATLAB and then are translated in the R language. Finally, also the model takes into account some laboratory setup information, such as the format of your well. It can be 96 to 24 because, of course, you know that the more the number, the smaller are the size of the well. So you have to take this into account, the radius and the size of the well, so the top radius, the bottom diameter, and the area. How much volume are you inputting in your well? The surface of the medium which is in contact with the medium, and the type of plastic. And of course, experimental conditions such as usually 37 degrees, the number of cells that are plated in your well, and the exposure time and the interval time. All this information needs to be given to the model in order to be able to simulate. This is after you have all this information, you can run, and this is an example of how the model is giving you output. So you have 4 different types of graphical representation. The first is the viability versus concentration. The second on the top right is the relative number of cells in time, so looking at the cell growth. Then on the bottom part we have the first on the left, which gives you the concentration in the medium versus time, and the second one, the most important, is the concentration, the intracellular concentration versus time. So what is the applicability of such a model? So we came out with 2 types of groups. For scientists, you can use them to go from a range of concentration to an intracellular concentration in order to refine your in vitro study. And for the risk assessors, you have, again, a range of concentration. You are able to achieve an intracellular concentration that can lead to an effect, and you can help yourself, or you can help to identify a point of departure in order to perform your starting point as in the risk assessment process. This is the list of chemicals for which we have established the so-called virtual cell-based assay. We have 30 chemicals. We have 4 types of cell lines, so cardiomyocytes for heart, HepG2 and HepRG for the liver, and A549 for the lungs. We then also look at the repeat exposure only in HepRG and only for a few of these compounds. I would like to just mention that all the information, if you would like more information on the background of this model, can also be downloaded from the Earl Ekman DBLM database, which is available at this link. And if you have more information on the DBLM, you can ask the scientific officer in charge, and you have her email at the bottom of this slide. Finally, if we put together how can we perform in vitro to in vivo extrapolation? We try to do this by putting together the 2 models that we just developed, and I would show you in this slide how the process was done. So you have the PBK model. We included inside the physiologically based kinetic model a new compartment which describes the cell, in order that you go from an external dose to the concentration reaching the target organ to the concentration in this target organ that goes inside the cell, in order to be able to compare on the other side the amount that enters in your well, that goes in intracellular concentration, so that you can actually compare these 2 concentrations and link the effect that is measured in vitro to the in vivo dose, as you can see in the bottom graph. We have published 1 paper, and here you can see from Gajewska et al. 2015, where we have written the dermal external doses, the concentration that reaches the liver, and the concentration in the cells. And these were all simulated by this type of model, because all of this was then implemented into KNIME in order to do it in an automated way, so you don't actually see this. It's just one workflow directly. You just input the dose, and then you get your output. And you can also see here the results. So you have a dose versus the effects, and the effects that were measured were cell viability and mitochondrial membrane potential. How can you use this graphical information, let's say, for extrapolation of in vitro to in vivo? You can go either forward from external dose to be able to extrapolate to your effect, or you can do reverse dosimetry, so go from the effect back to your dose. For this type of in vitro to in vivo extrapolation, we were able only to perform it for 5 chemicals, We had 5 PBK models and information on the vitro cell-based assay for Hep-RG and Hep-G2, so for liver cell lines. And with this, I would like to finish by saying that we have implemented all of this into NIME workflows. And to this after the coffee break, you will have a hands-on session, and I will explain more about the workflow themselves and how they were built. The final slide, just to conclude, is that I think we have achieved 2 nice biokinetic models which can hopefully be used in the regulatory process and the risk assessment process. And we will have also a special issue at the end of this year, probably available in early 2016, especially on the virtual cell-based assay in toxicology in vitro. Thank you very much.
Okay, thanks very much, Alicia. Any questions from the floor? Any online? Do we know?
Hello, Beate Nickel from IVIVE in Colworth. Your PBK model, you're in the process of developing a generic model so a user could actually populate it for their chemical of interest. I was interested, is the virtual cell-based assay model, are you planning the same? Because at the moment it seems like you can work with the 30 chemicals for which you have populated the model. But if you have if you're interested in a different model, in a different chemical, it doesn't seem to be possible to input your parameters in anything.
So the difference between the PBK model and the virtual cell-based assay is that the virtual cell-based assay was already built in order to be kind of general. Now we have only developed the NIME workflows for only these 30 chemicals. But if you would have all the users or all these parameters that I listed for these 4 type of submodels. So for the accuser, some, I don't know, log P's, the Henry constant, the really difficult one is, I think, the molar volume to achieve. There are some of these that are also difficult by accuser to achieve. We are trying to work on some like automated way. This is much easier for us to perform for a new call. While the PBK model is a bit more complex, let's say, to be more general. So the only part that then is needed is, in order to optimize, you need to have an in vitro response curve in a cell line. So you need to tell us like what type of cell line you are working. So it's more difficult actually to implement a new cell line in the VCBA than actually the chemical itself.
Generic model too.
No.
The way I understood your answer is that I could give my data to you.
No.
Or is the plan that there's going to be an interface where I can put in to go about generating the data to input?
So for now on the NIME web portal there is only available these 30 compounds before cell line. Of course, if you download the NIME desktop version, we can send you the VCBA. Okay, I have to ask the JRC people. This you implement and you will use. And with this special issue, we hope to cover every aspect because it will be around 7 papers only on this. So we really hope that then people can also use it and implement it and even try to make it a better model, refine it.
Thank you very much.